Our next speaker, who's Sohrab Shah, who's coming from Canada to talk today about some statistical models he's been developing for inference on structural variation in cancer genomes. Thank you very much.
Okay. Thank you, and thanks for inviting me to come here. Actually, I'll be talking about single nucleotide variants, just to make that point clear. As Peter very nicely demonstrated, sequencing of cancer genomes is going to yield a very compelling data set in the years to come. So currently in our databases that we have, for example, one is Cosmic, that actually stores and catalogs a list of somatic mutations. There are about 100,000 mutations that are in that database. These, of course, are mostly obtained through targeted studies. So an investigator has a gene of interest that they suspect might be involved in cancer, sequence a number of cases, find a mutation, enter it into the database, and publish it in the literature. But of course, next generation sequencing offers the capability to investigate these the instance of somatic mutations across the whole genome, and therefore we can interrogate these genomes for somatic mutations in a completely unbiased way. And currently, right now, there's a massive data generation activities that are taking place on part of the International Cancer Genome Consortium, the Cancer Genome Atlas Project, the Sanger Center. We in Vancouver are generating a number of these genomes, and literally we're poised to have in our collective community thousands of tumor genomes and transcriptomes, and it's really key. And again, I think Peter nicely showed that having analytical machinery is really necessary to gain knowledge from this data. Once we do this, it's almost certain that the mutational landscape as we know it will be completely redefined. And ultimately, we hope that this will lead to better understanding of tumor biology and eventually lead to better clinical tools for management of care. So by way of analogy, I think this is a map of the world circa 1600 when the explorers were going out and trying to map the continents, and they had a vague idea of where the continents were and what they looked like. And this is probably where we sit right now. But ultimately what we hope sequencing technology will give us is a really precise view of the mutational landscape in order to understand and further our knowledge of cancer biology. So I'm going to talk a little bit about how we model allelic counts with binomial mixtures. And yesterday we saw how the pre-processing of the data is subject to numerous errors and numerous biases. And so with that in mind, we're going to take the approach of taking the upstream perspective under the assumption that actually what is produced from the alignments and the base calling is actually useful information. So this is just a picture showing how reads align to a reference genome. And let me just see if I can. Okay. So here I'm showing, here's the reference sequence here. And I'm showing reads as they align, and they're ordered along the genome here. The black letters represent nucleotides that match the reference, and red letters represent nucleotides that convey a mismatch. So the way we approach is to take this alignment and collapse it into two numeric vectors, which store the counts of the number of reads that match the reference and the counts of the number of reads that convey a mismatch. Okay. And so as you can see that the singletons here are probably a result of either misalignment or sequencing error. But then when you get some evidence that there are multiple reads that support a variant, these would be positions of interest that you'd want to call. So the way we model this is with a probabilistic graphical model. And it's a simple binomial model where we represent the number of reads that match the reference with A sub I. The depth is the number of reads n sub I. And then what we want to infer is a genotype. So given the depth and the number of reads that match the reference, we want to ask the question of three possible genotypes, AA, AB, and BB, which was most likely to have given rise to the data. So we encode this with a probabilistic binomial distribution, and so we have a parameter that encodes a genotype-specific expectation for exceeding the reference. So, for example, the AA would be highly skewed. You expect that parameter to be very high. For heterozygous AB, you'd expect around 0.5, and for homozygous BB, you'd expect it to be very low. And then we have a prior over genotype. So this represents if you were to take all positions in the genome and randomly pick a position, how often would you expect a variant to occur? And this notation, by the way, is a probabilistic graphical model where the shaded nodes are observed quantities, and the unshaded nodes are the quantities we want to infer. So you'll note that both these parameters of the model are actually unshaded, and I'll explain how we actually infer those in a minute. So then finally we use hierarchical Bayes to regularize these estimates, and so mu is generated by a beta distribution, and pi is generated by a Dirichlet distribution. So that gives you an idea of the model. And so once we have this model, and let's say we have the parameters in hand, we can ask the question, given the model parameters, what is the probability that genotype K gave rise to the observed data at the position of interest? And this is just simply using Bayes' rule, and so you can see that this model, which we call SNV mix 1, is just a mixture of binomial distributions with component weights. Okay, so why is this advantageous? Well, when we first started doing this work 2 years ago now, the community was dead set on using thresholds. So I will not call anything unless I have, let's say, 10 reads at a given position. So we wanted to do some work, wanted to see if we could do better than that and see if actually we're thresholding out data if we did that. So as you can see here, so this is the posterior distribution of the 3 genotypes at different depths and different proportion of reference alleles. So as you're going this way, the depth is increasing, and so the y-axis here just shows the posterior probability. You can see that even at n equals 5, we can get fairly confident estimates, especially if the data are pure in one way or another, given this model. So we did an experiment where we took data from a breast cancer genome, and for which we had matched data from an affymetric SNP6 genotyping array. And we asked the question, if you were to eliminate all data at various thresholds from 0 through 10, what would happen to the accuracy? So we're taking the array as ground truth here, and that has its caveats, no question about it. But given that assumption, we did this experiment. And as you can see, that at thresholding at 10, this is an ROC curve, which is plotting true positive against false positive, right? The sensitivity is drastically affected by thresholding. And as you relax the threshold, the sensitivity increases progressively, but at no false positive penalty. And so the best curve we got was actually with no thresholding at all, and we just let the model tell us based on its assumptions. Okay. So I mentioned that we learn the parameters. So why do we do this? Well, there are a number of nice tools out there for discovering SNPs from normal genomes or diploid genomes, and these also have underlying in its structure a binomial distribution, but use fixed parameters that assume a normal genome. Cancer genomes, of course, are often not diploid, as we just saw. They're mixed in with normal cells, and they exhibit intratumoral heterogeneity. And so the parameters that assumed for a normal genome might not apply readily to a cancer genome. So we took the approach of fitting the model to data in order to potentially learn more representative parameters. So we just do this with EM, and I won't go into the details here. You can ask me later if you wish. And then we did an experiment. This is actually on 16 ovarian transcriptomes. Transcriptomes have their caveats as well, but as we saw yesterday, most of the data behaves properly. And so we took 16 ovarian transcriptomes, ovarian cancer transcriptomes, and this yielded about 150,000 positions for which we had matched affy SNP6 data. We did 10 repeats of a fourfold cross-validation to estimate parameters, and then which consisted of running EM on 3/4 of the data and then computing the estimate of the variance on the remaining. And when we did this and we compared to MAC, we got a significant increase in accuracy, and this is just by area under the ROC curve measurement. And this is published in Bioinformatics earlier this year. Yeah, I'm going to get to that in a minute. Yeah. Okay, so going beyond actually assuming that the base calls are correct, as we saw yesterday, there's uncertainty in that process. There's also uncertainty in the mapping or the alignment of the reads to the genome. So we can leverage this, and if you just focus your attention up here, it's hard to see this figure, but hopefully you can see this, is that we basically have two sources of uncertainty. We have the base call quality and we have the mapping quality. And so if both are low, we'd be less inclined to believe the data. But if both are high, we'd be much more inclined. So the higher contrast will yield more information. So what we did is decompose the binomial into a product of weighted Bernoullis, such that we were able to take in both the mapping and the base qualities into account. And this results in essentially a soft weighting of each read according to how confident the base call and the alignment was. So we did a little simulation, and this is simulated data, and so should be taken with a considerable grain of salt. But the idea is that if you see something enough times, even though it's not crystal clear, there's a suggestion that it would be there. And what we found is that at about 0.9% quality or confidence in the base call, the ROC curves really started to look very similar. So once you have enough evidence, the suggestion is that that's still useful data. And again, what people tend to do is to threshold out low quality base calls and throw that data away, and it might yet be useful in actually inferring SNVs. So when we computed the accuracy of this SNV mix 2 model, we got a small increase in accuracy, but most importantly, we got this without having to do any thresholding of the data whatsoever. So we then actually evaluated this on about 500 novel, non-synonymous single nucleotide variants that originally predicted using the SNV mix 1 model. And this is from this lobular breast cancer genome, and this is just at 10x. And when we validated these 500 using Sanger sequencing, we came up with about 305 true positives and about 192 false positives. So what we did is actually evaluate the accuracy on this ground truth of the other models, and found that the best accuracy we got was with we essentially got equivalent accuracy of using the SNV mix 2 model without having to threshold. And then we actually used a combined approach where we just thresholded the really bad data, just the data that was completely uninformative. We combined the two, we actually got a significant gain in accuracy here. So the best approach was to get rid of the really bad data and then do probabilistic soft weighting on the remainder. Okay, so that's just the statistical framework, but of course what we're interested in is applying this to actually learn something about cancer biology. So I just want to give you an example of what we did about this time last year. We were in the middle of analyzing this breast cancer genome. So the question's already come up, is if you compare a primary tumor and its metastasis, what do you see? Well, when we were planning this project, we identified a tumor in our tumor bank for which we had the primary tumor and the metastatic tumor from 9 years later. And this is an ER positive grade 2 lobular carcinoma. And the results of this were published in Nature on October. And we had the question, we wanted to ask the question, first of all, how many mutations were present in the metastatic tumor to begin with? And then we wanted to, because we had the primary tumor in hand, we can ask the question of how many new mutations arose over time. And then finally, can we use the allelic distributions that we see in the data to detect mutational heterogeneity? So we generated a large data set, 3 billion paired end reads, or about 120 gigabases of a line sequence. And we also had the transcriptome for the metastatic tumor, and we generated 6 gigabases of a line sequence for the transcriptome. So the results of this were that with the joint analysis of SNV mix on the transcriptome and the genome, we got about 1,500 non-synonymous variants were predicted. 1,200 of these were sent for primer design. Another 1,100 or so were actually survived that process and were sent for Sanger resequencing. So to our surprise and much to our chagrin, that actually only about 440 were confirmed. And there are a number of sources for this, which I'll get to in a minute. So, but after looking at the normal DNA as well, we were able to confirm that 32 of these were indeed somatic. And when we compared these, looked for these 32 somatic mutations in the primary tumor, we found another somewhat surprising result. We found that 19 of these were actually completely absent. They just weren't there at all in the primary tumor. We found that 6 were present in the tumor, but at low frequency, and I'll show you that data in a minute. And that 5 were prevalent at high frequency in the tumor. This has implications because it severely narrows down the scope of which mutations were actually the candidate driver tumorigenic events. We then looked at some of these mutations in 192 other cancers, 112 lobular and 80 non-lobular, and found that in 0 out of none of the cases actually had any of the same mutations present at the same position. 3 cases had nearby mutations in HER2, and 2 had truncating variants in a gene called House3. There were some cases that had mutations again, but not at the same positions that were present in cosmic. So these genes are mutated in other cancers, and in breast cancer as well, but the actual precise mutation that we saw were not present in the database. None of the genes that we found mutated were in common with the CAN gene surveyed from the ER-negative breast cancers from the Vogelstein work. So the point is that most of these mutations were completely new and occurred in genes that were just never known to be previously mutated in breast cancer before. So I'll just spend a bit of time explaining how we interrogated the primary tumor. So the primary tumor that we had was the source of the DNA was from formalin-fixed, paraffin embedded, and we actually tried to sequence this genome using high Illumina sequencing using whole genome shotgun. The data was not as good quality, and so we were a little bit reticent to use it directly. And so what we did is we took a targeted approach where we amplified, we designed primers around each of the 32 somatic mutations, and just actually quite short amplicons. There's about 100 to 150 base pair amplicons. Amplified them up, pooled them, and then sequenced them on the Illumina GA2. And we literally achieved about a 10,000 to 50,000 fold redundancy at each one of these positions. And then what we did is actually compute the background distribution. So just to get an estimate of what the error rate would be around the variant of interest, we computed this background distribution. And then we were able to ask the question of the data, do the variant positions in the primary have more non-reference alleles or variant alleles than expected by that background distribution, just with a simple one-tailed binomial exact test. So the results were as follows. So this is actually representing just 28 of the mutations for which we could actually get this reaction to go. So we found that 4 of the cases, so what I'm showing you here, is the ratio of variant allele to wild type allele in the primary, and over here is the same ratio but in the metastasis. And you can see that we had 4 mutations that were about 23% or more of the alleles were present in the primary. We had another class for which we had only between about just under 1% and about 14% that were present in the primary. So this suggests immediately that at the time of diagnosis in the primary tumor, there's already significant mutational heterogeneity and clonal populations that existed in the primary. And then the rest were just vanishingly rare. You can see that these are basically all 0. As a positive controls, we did the same experiment in the MET, and all of them were present at reasonably high frequency. Okay, so I mentioned that we actually sequence a transcriptome as well. And here's where the probabilistic model really comes into play. We noticed that there were some variants that were in the transcriptome that just didn't validate at all in the Sanger sequencing. And all the validation was done on the DNA. So we might have predicted variants in the RNA-seq, but then we did the validation in the DNA. We noticed that a number of these didn't show up. And so we thought that, well, maybe they're actually there, but they're there in the RNA, but maybe they're just not. They really truly are not present in the DNA, and this is these variants then would be post-transcriptionally modified, so they're actual RNA edits. So this is a known mechanism of mutating or altering the RNA, and it's been known for pathogens to use this mechanism to evade the immune system. And so we took a number of positions for which we had high confidence of an SNV in the RNA and a high confidence of being wild type in the DNA, and then went and looked at these and validated quite a number of them by Sanger sequencing. In particular, 2 of these, a variant in COG3 and SRP9, actually would be predicted to induce an amino acid change. So this is a post-transcriptional event that will induce an amino acid change in the protein. Here's just an example of the validation data for these 2 events. So you can see here in the tumor DNA, there's basically no evidence of the variant whatsoever. And in the RNA, there it is, nice and present. Okay. So after doing this work and after the paper came out, we actually went back and looked at a few more. And we actually found that we found 13 additional coding edits that were confirmed. Okay, so after we'd finished that work, I started wondering about some of the events that Peter was talking about. What is the effect of copy number on allelic distributions? And so we designed a model that can essentially integrate copy number alterations into SNV analysis, and found that it indeed revealed somatic mutations. So this can really be considered as a second pass analysis of this genome. And just to illustrate the concept, up here I'm showing a SNP array of the normal DNA from this particular individual. And you can see nicely that the B allele log ratio shows the expected 3 bands of showing the 2 homozygous alleles and then the heterozygous positions in the middle. This is the array from the tumor, and you can see that this nice pattern that we see here is significantly altered. And what I'm showing here is the results of analysis to do segmentation on this array. And the red bits are just showing regions that are predicted to be amplified. And what this data suggests strongly is that this is an allele-specific amplification that is skewing the allelic distributions of all the heterozygous positions within. This down here is showing the same data but from the sequencing data. So we're able to predict copy number from the sequencing data, and again, blue regions are neutral, red regions are highly amplified, and the allelic ratio suggests that there's really nothing in the middle anymore. It's all been skewed away. But this is not, the important thing to note here is that this is not loss of heterozygosity, this is just allelic skewing. The other allele is still present, it's just that one allele has been preferentially amplified. So to answer your question, basically what we did is we took the simple step of just expanding the genotype state space according to copy number, and where if there's, and really this is an approximation of copy number. We just used 5 discrete states. It's a crude approximation, admittedly, but we just wanted to see what we could learn from this. And again, we expand the state space as follows. and up to high-level amplification where we get 6 possible genotype states. And what we do is we basically chop the genome up into these different discrete copy number groups, and then fit the binomial mixture independently according to the expanded state space to each one of those regions. So I'll just skip over the details of this, but the concept is here is basically that in the diploid region you have your AA, AB, and BB, and in amplification regions you expand the state space. And here I'm just adding this other node, this observed node. This model requires that you know the copy number ahead of time as input, and then accordingly dynamically alter the genotype state space. So when we did this and applied it to the same genome, that same tumor, breast cancer tumor genome that I just described, we found that, so this model is called Conan SNV, copy number annotated SNV. Compared to MAC, we compared to our diploid mixture model, we found that there was an enrichment for non-overlapping variants in the Conan SNV model. So we took these and we projected these onto protein coding space and picked, there were 200 actually, we picked all of them, all 200 non-synonymous protein coding variants that were unique to the Conan model, and used the same targeted ultra-deep amplicon sequencing that we used in the previous experiment that I showed you earlier. And the validation results were as follows. We had 76 that just failed PCR. This was, again, not an undesirable failure rate, but nonetheless, we pressed on. 47 false positives, 43 were predicted as germline, but very surprisingly, 24 new somatic variants predicted by this method, almost effectively doubling the number of somatic mutations in this genome. So here's what the data look like. Here's the normal, and you can see that this is almost all 0. It's the same type of data that I showed you before. And here's the MET, and basically the average allelic ratio here is about 17%. So these are all variants that were not detectable by the diploid method, and you can see why. It's because the skewing of the allelic ratios is highly towards the reference. So you can ask the question, does this induce a higher false positive rate? We check this against using OncoSNP actually, and on the array. We found that in the lower amplification regions in neutral, there's basically no difference in ROC curves. And then finally here in the high level amps, that's where we found the most difference. And indeed, the Conan model was quite a bit more accurate. So I think I better stop there and just in conclusion, just state that we found 56 novel somatic mutations in the metastatic tumor. 50 of these were in genes that were not known to be mutated in cancer. I think the point was raised yesterday that this is an example of one of those n=1 experiments. We need to do more of this. The comparison with the primary tumor revealed significant mutational evolution and intratumoral heterogeneity. And then finally, the integration of the copy number into the SNV analysis effectively doubled the number of validated somatic mutations in this tumor. And I think I will finish there because I'm out of time. I just wanted to thank Sam Aparicio and David Huntsman and the people at the Genome Sciences Center down the street in Vancouver who generated all the data. And finally, all the courageous patients who donated their specimens to research, without whom we couldn't really do this work, and my funders. So thanks for your attention.
Time for a couple of questions. Yes?
The idea of the threshold-less analysis is of course important, but in the end you combine some thresholds with your analysis. What kind of thresholds did you
So we just use the really extreme points of the distribution. So essentially anything that, so we actually looked at this. We combined all possible thresholds or reasonable thresholds in the paper, and we found that the ones that were optimal were to really be stringent on the, so be permissive on the mapping quality. So we only filtered out the other way around. So we only kept the really high quality alignments, but with the base quality, we're able to be really quite relaxed with the threshold. Yes?
What determines, so the two talks focused, it seemed, quite differently on copy number changes versus some point changes.
changes.
So how important is it to combine them? And do the two have different roles in different cancers, or is it just a matter that doing it all together is too complicated, so we're starting off right?
Yeah, I think so. The answer to both questions, yes. So in different cancers, so some cancers are quite genomically stable, and so copy number doesn't play nearly as much of a role. We found a defining point mutation in granulosa cell tumors of the ovary, which are a subtype of ovarian cancer that are really quite genomically quiet. And this is a case where literally this disease is now defined by this mutation. We pulled in about 100 cases from all around the world because it's quite rare. We have to call in favors, and really we were able to find this mutation in 97%, the exact same point mutation, and essentially it defines the cancer. But in breast cancer and lung cancer, in high-grade serous ovarian cancer, these are highly genomically unstable events. They're characterized by mutations in P53 or BRCA1 and 2, and so it really depends on the disease itself. And cancer is one disease is probably never going to work. It's a set of different diseases. Yeah.
Okay, I think we've got to leave it there. Thank you.